Good morning and welcome to the Spatial Genomics workshop. And my name is Jyoti Verma. I'm a field application scientist from Decode Science, and I will be hosting this workshop. So before we get into the agenda of this workshop, I would like to mention few things about Decode Science. So we are a distributor company, and we supply BGI products in Australia and New Zealand. And besides BGI and MGI, we also supply all these high quality, cutting edge genomics brands, including single cell sequencing kits from Pars Bioscience and high-C assay kits from Dovetail. And you can find some of the literature about these products in the MGI bag, which we have provided. And besides this, we also supply and provide technical and application support for all these brands. And we have an excellent sales team, and our business development managers are based in different cities in Australia and New Zealand. So even in these unprecedented times and when border lockdowns happen, we are still able to provide excellent local support to our customers. And if you have not got chance to get into our booth, please come today afternoon and or visit our website. So coming to the outline of today's program, so we have three 15-minute talk followed by 5 minutes Q&A session. So first, Dr. Kira Sun will provide introduction to BGI latest innovation in spatial omics, STOmics. And then we have two talks from our early access customers in Australia, highlighting their discoveries made using BGI STOmics technology. So bit about Kira, introduction. Dr. Kira Sun is a BGI field application scientist supporting the application of BGI spatial omics innovation in Australia and New Zealand. She joined BGI Research, a non-profit scientific research institute as a senior researcher in 2017 after completing her PhD in Ghent University, Belgium. She has a background in development of novel high-throughput metagenomic sequencing technology of diverse microbial samples. And in her talk, she will focus on BGI STOmics technology and how BGI technology has overcome previous spatial omics technology and provide nanoscale subcellular resolution of 500 nanometer. Over to you, Kira.
Thanks for the introduction from Jyoti, and thank you all to getting up so early and to join the workshop today. And I'm excited here to introduce you about our new technology on transcriptome, on spatial transcriptome profiling. So first of all, I would like to introduce the BGI group. So BGI provides a wide range of NGS services and genetics tests to advance life science. Under BGI group, there's non-profit institute like the BGI Research, where I'm come from, and the Giga Science Press. And we also have the commercial subsidiaries, including the BGI Genomics and MGI. So BGI now present globally in over 100 countries and regions. In Australia, our colleagues are stationed at many places, including Melbourne, Sydney, and Brisbane. BGI Australia has established from the year 2016 in Brisbane. Then a state-of-the-art sequencing laboratory was opened in QIMR. Last year, the MGI Australia incorporated and opened a demo lab in Brisbane as well. So to give you a brief introduction of the MGI demo lab, and it showcases ultra-high, high, and medium sequencing throughput DNB sequencers, including the DNB G400 and the T7, which can produce 1 to 6 terabytes sequencing data per day. And our demo lab also presents automatic laboratory systems like the MGI SP series and bioinformatic products to build on the existing technologies. The demo lab now provides support to local and sub-regional users to accelerating their research and applications. So back to topic, I would like to focus on our new technology, which is developed by BGI Research, called Spatial Enhanced Resolution Omics Sequencing. The approach allows transcriptome profiling of large histological sections with high resolution and sensitivity. So we now have this technology for scientific trial and is branded as STOmics. As you all know, organs are the function unit of human, and these come from complex heterogeneous tissue. So the previous technologies and bulk or single cell RNA sequencing could not provide information of tissue architecture, which will lose the information of position-directed cell fate or cell-to-cell regulations and so on. However, the spatial profiling of gene expression enables providing a complete picture from an organ level to a single cell level. So it's offering more biological information, including all different cell types in the context of heterogeneous tissue and the molecular complexity underneath the tissue morphology. First of all, the factors that affect the spatial profiling resolution is the spot size and the distance between the spots. So if those sizes are too big, it will reduce the capture resolution. The idea of StereoSeq comes from DNA nanoball sequencing platform. So on the modified silicon surface, we deposit DNA nanoball containing the coordinate information. The coordinate information deposit into the oligo sequences, which will link with our tissue mRNA later on. The space between the DNA nanoball spot is 500 nanometer from center to center, and each spot size is about 220 nanometer in diameter. So this means for mammalian cell, it's usually 10 micrometer in diameter, and it will contain 400 spots to capture the mRNA inside of the cell. So our StereoSeq is a technology based on the in situ capture method. Basically, what we do is we first sequence the DNA nanoball to get the coordinate information, and then we link the UMI and poly-T to the DNA nanoball, and those goes with the chip that's supplied in by our company. And then during the experiment, what you may do is you do the tissue sections from the cryo state, and then you lay the tissues on the chip. The chip will capture the mRNAs from the tissue spatially. After that, the captured mRNA will follow the process of reverse transcription and library preparation. In the end, all the mRNAs from one chip will be put into one tube for sequencing. And this experiment will last 1.5 days to finish. So our chips comes in a variety of sizes, and we can customize them for different tissues. In this way, we can carry out the whole tissue profiling that has never done in this way before. So such as large tissues from monkeys or human organs. And the 1 centimeter multiply 1 centimeter chip is already available right now. I think we have a prototype here and to show around, and which is already big enough for most users. One of the advances of our technology is having the largest field of view and the finest resolution. So using the stereo-seq, it will feel like overlook a specific street or a specific house from the space by Google Map, while still keep the image of the Earth. This requires 120,000 times reduced vision to such precise level. There are many spatial profiling technologies currently. So compared to the existing technologies, the stereo-seq can reach to nanoscale spot size and distances. So meanwhile, maintaining the ultra-high capture region. So to give you a better understanding of our technology, that we use tissue like mouse or factory bulb and brain. Those are the widely used tissues for spatial transcriptome profiling. And so we also have tested those tissues. So the capture rate has been evaluated comparing the published data. And from the bar chart, that you can see the comparisons, the orange bars represents the stereo-seq. So for instance, in a 10 micrometer diameter area, the median 550 UMIs were captured by SLISEq V2. From mouse embryo sections, 848 UMIs on average were captured by 6-scope. However, median 1,450 UMIs are captured by the stereo-seq. So generally, stereo-seq can capture more UMIs in the tissue sections. The resolution of different technologies can be more intuitively displayed through the detected MRA map. So the first two rows represents the expression of gene markers captured by stereo-seq. And the third row is the gene markers captured by HDST, and the 4 rows is captured by SLISEq V2. And the last row, and the last row represents an image generated from the in situ hybridization. So the distributions of several specific markers captured by stereo-seq exhibit remarkable similarity to the in situ hybridization and give you a full pictures of their expression in a tissue level. So furthermore, the cellular resolutions in adult mouse brain can be reached by our technology. So like in the middle, the red dot shows the detected MRA by stereo-seq, and the blue dot are the MRAs, remains the cells has been stained by a nucleic acid dye stained MRA. So each blue dot refers to one single cell, and the captured MRA is highly overlapped with the RNA staining image. That's meaning the stereo-seq can really capture transcript at cellular resolution. In addition, that we have released several bioinformatic tools to support the analysis of the data from stereo-seq. So stereo-seq analysis workflow now is already released on GitHub. So it can support the analysis from the beginning. When you obtain the sequencing results, it will provide tools to do the mapping of the sequencing according to the coordinate information on the chip. And then you can do tissue register, tissue cut, saturation analysis, and so on. So another online tool is called StereoMap, so which allows you to visualize the sequencing data by selecting the whole tissue sections or the specific regions that you can provide the transcript clustering and statistics. So here, the figures in this slide are the results from the StereoMap. The details of STEROSEQ technology has been submitted, but the preprints are available online now. So including the studies on mouse embryogenesis, the whole developing stages from the implanted embryos and after the implanted embryos has been used by our technology, and the molecular basis of the regional specification and neuromigragation and differentiation in tissues overall, this developmental embryos has been studied. We also have studied the Drosophila embryo and larva development, some tissues from the liver tumor, and also the development of the zebrafish embryogenesis, and the extraordinary regeneration ability of the axolotl brain upon injury, and also the structures of the Arabidopsis leaf. So indeed, using our technology, it can not only apply to animal tissues, it can also apply to the plant tissues, expanding our knowledge on nature. We have performed in collaborations with a lot of institutes of this technology on several organs or tissues of mouse as a model animal. And besides it, so we actually do a lot of different organs. And besides that, we had also experiment on the zebrafish I showed slide before, and amphibia and other organs. The STEROSEQ is able to provide large field of view, and it can also provide the microstructures, as I showed from the figure here. So you can clearly see the epidermis and dermis on the lip of developing mouse at the stage of E50.5 during embryogenesis, so which requires high resolution. And this figure is come from the unsupervised clustering of the mRNAs captured by this technology. So another point that I would like to stress here is, as the STEROSEQ signals are very dense, and for some tissues, the continuities between the cells adjacent is very good. So we can study the cell microenvironment, cell migration, and the cell migration trajectory. In the end, I would like to acknowledge the whole STOmics team for their innovation, and we have collaborations with so many institutes globally now in this technology. In Australia, we have collaborations with Professor Ankur Sharman and also Dan from Peter Mac, who will present later. And we also have collaborations with University of Queensland and so on. We have initiated mailing collaborations in Australia. So BGI is in the process of forming the Spatial Temporal Omics Consortium. And if you are interested in touch, and we are gladly to hear from you. So we hope by our efforts that we can make this technology more affordable and accessible for many users. And I would like to thank you for your attention. And I'm ready for questions. Thank you.
Thank you. It's truly lovely work, and I'm excited to hear more about it and try this out myself. But the question I wanted to ask was, you didn't actually show subcellular localization, for example, in the development, maybe from the oocyte or very earliest stages of the embryo.
Yes.
And I was wondering, how much can you zoom in there, and how much detail do you get at the subcellular level?
At the subcellular level, you mean?
Yeah.
I think you can. Maybe you can give me an example of what kind of tissues, because it's different from tissue to tissue. It's like for the mouse embryogenesis, the capture rate is super high and the density is good, so we can get a lot of information on that. Yeah.
Right. Yeah, I guess the experiment that I would love to do would be in the mouse oocyte, where you have localized RNA and then trying to correlate RNA localization with translation, for example, where RNA is sequestered during embryo development, but then released from granules. And I was wondering if you would be able to see that level of subcellular localization change over development.
Yeah, I think so. Yeah.
Okay, amazing. Thank you.
Thank you.
So I just want to add, theoretically 500 nanometer is possible, and we have spots there. So you can get that resolution if you sequence.
Hello. Does this work? Oh, sorry. I maybe missed it in the slide, but you had a big table that showed comparisons with your technology and the other technology.
Oh, sorry.
And I believe that this is only applicable in frozen sections. Is it possible that it would work in FFPE tissues as well, or is that something that you're still developing?
So this technology applies to frozen tissues or the fresh tissues embedded by OCT. So for FFPE, it can be supported by other technologies.
Okay, thanks.
Yeah.
And we have plans to do FFPE. We are working on it, but currently we only do fresh frozen. If there are no more questions, I don't see anything here.
Okay.
Thank you, Kira, for lovely talk.
Thank you.
So our next speaker today will be joining virtually, Dr. Dane Vassiliadis is a postdoctoral fellow in the laboratory of Professor Mark Dawson at Peter Max Cancer Center, Melbourne. He studies the role of epigenetic factors in the control of gene expression, and recently he has developed cutting-edge cellular barcoding methodologies to investigate the role of epigenetic priming and adaptive processes playing on initiation, maintenance, and evolutions of cancer, such as acute myeloid leukemia, particularly in response to therapy. So in his talk, he will talk around intra-tumor heterogeneity using the new BGI STOmics technology. And so welcome to Dane virtually.
Hi, everyone. Really nice to be joining you virtually. I wish I could be there in Lorne. It looks like it's been a fantastic conference so far. Thanks for coming out this morning to see some of this really early work that we've been doing at Peter Mac using the STOmics platform. I just start by acknowledging, I guess, the traditional owners of the land on which I'm broadcasting from, the Wurundjeri people of the Kulin Nation. I want to pay my respects to their elders past, present, and emerging. I want to thank BGI and Decode for inviting me here today. I'll be chatting about some of our really preliminary work, looking at the spatial and transcriptomic heterogeneity of malignant clones within the leukemic niche. And I suppose, as way of background, we work on the process of intratumoral heterogeneity. Tumors, throughout the disease course undergo a functional diversification, which results in various tumor subclones that have different properties. Some will eventually seed metastases, and some will be primed to, relapse post-therapy. And traditionally, I suppose this was thought to be primarily due to the continual acquisition of genetic variants. And so this is a classic, genetic evolution model. But what we now know is that there's several examples in, a range of cancer types of non-genetic evolution models that exist primarily through a transcriptional priming or plasticity events that result in distinct functional outcomes for various clones. So we focus on acute myeloid leukemia, and AML is really a good example of a cancer that is incredibly genetically stable, and a particular subtype that we focus on, the MLL rearranged AMLs, can really be just driven by the single gene fusion event involving the KMT2A gene. Now, importantly, what we find is that while patients regularly achieve remission with standard chemotherapy and even some new exciting targeted therapies that have sort of come through the clinical pipeline recently, up to 40% in some cohorts, if you look at sort of pre and post relapse genomic sequencing, will show no evidence of new mutations, suggesting a non-genetic mode of tumor evolution. So, as I said, there's been over the last decade a plethora of papers now looking into and studying these non-genetic modes of tumor evolution and how they can result in therapeutic resistance and tumor progression. But ultimately, what we still lack a clear understanding of is the transcriptional programs that result in these outcomes, and I guess whether they're pre-existing before you apply some sort of selective pressure, such as a therapy, or whether they're adapted over the treatment time course. So we recently published a paper looking at exactly this, and I guess one of the challenges in understanding these questions is that we really lacked an ability to link the functional output of particular cancer clones to an underlying transcriptional or epigenetic profile. So we developed a system that would allow us to do this, and that system is dubbed Splinter for single-cell profiling and lineage tracing using express barcodes. So Splinter is really a lentiviral platform that incorporates a standard fluorescent reporter, but downstream of that, incorporated just ahead of the 3' UTR, is a synthetic barcode. And so that is actually incorporated into the transcript. And so these barcodes can be read out not only in the bulk genomic DNA, if you wanted to do sort of bulk profiling of a clonal profile, but also in standard single-cell methods that utilize 3' capture. So 10x or dropseq, or really anything that is using a 3' capture should be applicable using Splinter. And so using this technology, what you can do is actually link not only the lineage of particular clones, but also link that to transcriptomic profiles at the single cell level at various time points across your experiment. And so what we had done in our paper is take barcoded cells, expand them and transplant across multiple mice. So we're basically transplanting the same repertoire of clones into multiple mice, sequence the clones that drive disease, and then look back at this baseline time point to understand, using single-cell RNA sequencing and capturing the barcodes in the single-cell RNA-seq, look back at what transcriptional profiles drove that. And so what we found quite strikingly was that when you look across multiple mice that get the same repertoire of clones, there's near complete concordance in the representation and frequency of the clones that drive the disease. And I should just mention that this was from basically isogenic cells starting out. So there's clearly some sort of cell intrinsic priming going on in a non-genetic fashion. And then when we utilized Splinter to look at the transcriptional profiles, what we found was that invariably most clones had upregulation of secretory leukocyte peptidase inhibitor, a gene that hasn't been previously described in leukemia, acute leukemia, but more importantly, downregulation of a host of immune markers, particularly antigen processing and presentation type genes such as beta-2M. So we partnered then with BGI. In particular, we wanted to utilize the STOmics platform to now understand the kinetics of how these leukemic clones are engrafting and proliferating within the leukemic niches, namely the bone marrow and spleen. And so we had an experiment actually where we transplanted these clones into NSG mice, harvested bone marrow and spleen from diseased mice. We basically cut the tissues in half and sequenced half. So we got single-cell RNA sequencing from half the tissue, and then the other half we utilized for the STOmics platform. And so what we found, the single-cell RNA at the baseline time point There's a certain clone 52804, which was relatively lowly abundant in the isogenic pool of cells at the start, but then basically in all of the mice that we looked at, this clone expanded to incorporating at least 60% of the disease burden. So a massively dominant clone. And so we really wanted to understand the kinetics of how this clone managed to expand within the various niches. So we supplied fresh frozen tissue. In hindsight, we probably, as Kira mentioned, OCT embedding is probably the preferred step for supplying fresh tissue. We had just flash frozen them in liquid nitrogen, and nevertheless, what we saw was that we're getting still really good RNA integrity scores. So just a little tidbit for someone who might be thinking of doing this on some flash frozen tissue that they also have, at least for these spleen samples that we supplied, it seemed to work quite well. And then, comparing a healthy mouse spleen, you can see that we recapitulate the various features of the spleen. We've supplied cross sections, and you can see really clearly on this H&E stain, the white and red pulp, the lymphocyte containing areas, and the region where the red pulp where the blood is coming through and being filtered. That sort of translates into a nice gene expression map, and then through unsupervised clustering, you can see that most of those features are the resolution on those features are pretty well maintained with the STOmics data. Interestingly, we look at what we can also do with that high resolution data is actually pick out known features, known markers of the spleen. So, hemoglobin is marking the red pulp. There's various markers of these white pulp regions, and then also really nicely, this Marco gene actually marks the marginal zone that separates the white and red pulp, and that you can see that coming out quite nicely. In contrast, what we found that was quite interesting was that in the leukemic spleen, the morphology is severely perturbed. We're really losing a lot of that substructure in the H&E stain, which is perhaps unsurprising given that these mice were incredibly, they had an incredible disease burden by the time these spleens were harvested. We can see that most of the gene expression profile is taken up by this region up here, which kind of forms a primary couple of clusters in the unsupervised clustering, suggesting that this is probably where the leukemic cells are sitting. We can't actually get as high a resolution on those normal morphological markers, presumably because of destruction of the niche. There's still a little bit of marking of the red pulp regions through hemoglobin, but definitely, and some B cell markers as well. But definitely in this leukemic spleen, what we can see is that the morphology of the spleen and that substructure has been perturbed. And then when you actually look for known leukemic cell markers, what you find is that, yes, definitely these are myeloid leukemias. They highly overexpress MPO. So that's suggestive of where we've got engraftment and expansion of these leukemic clones, and that's sort of backed up by HOXA9. Nicely, what I talked about before was upregulation of this gene SLPI in these leukemic clones, and that's really what we see in this top right section map, and also relative downregulation of beta-2M as well. So we're kind of recapitulating with the STOmics platform all of the features that we had found in our previous work, which was really nice. So I guess most importantly for us was to try and understand whether splinter barcodes can actually be detected in the STOmics data. And as I said, this should be theoretically possible because STOmics is, again, a 3' capture method. So I think as Kira alluded to earlier, you've got coordinate IDs and a UMI ID with a poly T hanging off the chip. So you're going to be capturing transcripts via the poly A tail. And in our splinter barcode transcripts, essentially, that's exactly what we've got. We've got the splinter barcode sort of hanging out at the 3' end of the transcript. Ideally, what would happen is that this would be incorporated into the library and be able to be read out in read2 through the paired end sequencing of the library. And so I guess the most basic thing that you can do is look in the raw data. And encouragingly, what we found is that, yes, you can actually find a whole host of reads in the STOmics data that contain these splinter barcodes, which is really encouraging for us. But more excitingly, I think, was when we actually mapped the location of each splinter barcode to an individual coordinate on the section map. So again, just bringing back this leukemic spleen from before, I've kind of highlighted these two main regions of clustering from the unbiased clustering that we did. And then when you map these barcodes, again, remembering that 52804 was about 60% of the disease burden, that's this clone in red. And then I've just binned all other minor clones that grew out in blue here. And what you can see is that these clones form really distinct clonal territories. Perhaps not super surprising if you're a person that sort of works on hematological malignancies, and given what we think happens when leukemic clones engraft in the spleen. But what was more exciting was that when you actually look at the transcriptional profiles of these two groups of clones, they're essentially transcriptionally distinct, suggesting that these clonal territories are not just spatially defined, but also transcriptionally defined. Of course, you can do, sort of standard differential expression. And what you find is that this clone 52804 that has sort of exploded in the spleen, most of the upregulated genes are around growth pathways, which is unsurprising, but primarily in the downregulated gene fraction, what you find is markers that I guess would promote a more mature leukemic blast cell state, suggesting that this clone 52804 is able to either shield itself somehow from the immune system and or maintain a more, I guess, leukemic stem cell-like signature. And so we're really keen to follow this up in future work. You can see really nicely if you actually look at some of these marker genes that there's really clear patterning here for these clonal territories, whereas what we found in our paper, SLIPI, is basically overexpressed in both territories, and beta-2M is reduced in both territories as well. So I suppose the take-home messages are that, BGI STOmics, offers really high resolution data. You can clearly see various tissue and niche-specific effects and markers. We found, I guess, what we found with the leukemic clone-specific territories within the spleen makes us quite excited to expand and try this in new tissues, and we're currently trying some bone marrow from the same mouse. Obviously a much more challenging tissue to work with, and it's something that we'll hopefully get some nice data on soon. But more importantly, I think the power of this high resolution technology to find specific transcriptional profiles of various clones and within these clone territories really sort of hammers home what you can do with the technology. And I suppose we're really excited to delve more into this data. We've really just scratched the surface on this at the moment. So yeah, I think I'll finish there. I think I'm out of time. So yeah, obviously just want to thank BGI and Decode, particularly Azadeh from Decode, who's facilitated basically this entire collaboration along with Bi Cheng at BGI. So thank you, and I'll take any questions now.
Thanks. That was just really lovely. I was wanting to ask you, can you tell now the question you sort of opened with, which is what is the heterogeneity? And so you can see the two different populations, but can you see infiltration and what the cells surrounding those tumor clones, how different they might be?
Yeah, it's an excellent question, Trouter. So that's something I'm trying to actively work on at the moment. I think one of the things I've learned with STOmics, and kind of alluding to your question after Kira's talk about looking at subcellular RNAs, the sequencing depth that you acquire is very important. You can obviously appreciate if you're trying to look at spots that are 500 nanometers apart, individual spots, you need pretty high depth to capture, a range of transcripts for each of those spots. I think what we've had to do to date, and most of the examples of STOmics data that I've seen have binned the data somewhat. So you're sort of binning spots together. Definitely, I think it will be possible to classify various cell types that are infiltrating or surrounding our leukemic clones. I think for our data, we may need a bit more sequencing depth, but definitely it is possible, and it's something we're looking at doing in the near term.
Oh, it's really exciting. Thank you.
Any more questions from audience? Okay. So thank you, Dane, for a wonderful talk. And let's give him a hand. So our next speaker will also join virtually, Dr. Ankur Sharma. He is a lab head of Oncofetal Ecosystem Laboratory at Harry Perkins Institute of Medical Research and Women's Cancer Senior Fellow at Curtin Medical School, Perth, Western Australia. His research is directed towards exploring the similarities between fetal and tumor microenvironment. He is specifically interested in understanding oncofetal reprogramming of the tumor ecosystem. He is a member of multidisciplinary human cell atlas team to build the liver atlas from development to disease and liver cancer collaborative Western Australia. Ankur is among the first member of the 10x Genomics Visium Clinical Translational Research Network team, and he is also early access user of BGI Spatial STOmics platform. So in his talk, he will share his recent work on oncofetal ecosystem in hepatocellular carcinoma and its spatial localization based on the data generated from BGI STOmics platform. And the paper is under review in a top scientific journal. So let's hear from Ankur. Welcome, Ankur. And
Thank you. I hope you can hear me. First of all, thanks for inviting me for this fabulous workshop. And I wish if I could come in person, but as you know, it's very difficult to break the Great Wall of Western Australia. So in today's presentation, I'll be talking about our recent work in the oncofetal ecosystem, and mainly its spatial localization. I have another talk in the afternoon session at the Lorne Genome meeting, and some of my slides might be repeating, but in that talk, I'll focus more on clinical implication and spatial localization. So just to give you a brief, what is oncofetal? So oncofetal essentially means something which is shared between a fetal tissue and tumor, but absent during normal homeostasis. And as all of us know, ecosystem is a microenvironment. In terms of tumor, in our case, we are looking into immune and stromal microenvironment. And our lab studies similarities between embryogenesis, regeneration, and cancer. Now, looking into a tumor ecosystem. So I think I got fascinated about tumor ecosystem when I was studying tumor evolution like 3 or 4 years ago, how tumor cells evolve under various therapy response and all. And as Dan beautifully showed that how the different clones, they co-evolve with each other. And my fascination started with this process in terms of understanding when we give these therapy to tumor cells, what happened to the microenvironment? How do these cells evolve in response to changing dynamics of the tumor cells? And how does this reciprocal interaction lead to the evolution of the tumor ecosystem in terms? So if you look at any static image of a tumor, for example, in this case, this tumor tissue, you will see that there is a kind of a tug of war happening between normal tissue and tumor. So the upper part is a normal tissue and lower part is the tumor, and these cells are stained epithelial as green, immune as red, and stroma as yellow. And if you see, and if you envisage a tumor evolution from normal tissue, a mutant cell may have started the process of colonization, we see that there's a different part of the tumor have very different this a geographical localization. Specifically looking into this boundary of the normal tumor, we find that some of these immune cells, they are trying to infiltrate the tumor, while another part of the tumor, you will see this very red immune cells look like a very nice structure of like kind of blood vessels, which now these immune cells can infiltrate the tumor, but another part of the tumor, immune cells are unable to infiltrate, which means there might be two different clones of the tumor, which one clone allow immune cells infiltration, another clone does not allow immune cell infiltration. And I think this understanding is very important in terms of how immunotherapy may or may not work in a patient. If you zoom into some of these areas, so this is just a normal as you can get in a tumor tissue, and then this is a one part of the tumor where you see there's a change in the structure of the epithelial cells, but immune cells has not and are on the boundary of this tissue. Now, if you look into the another part of the tumor where like immune cells are unable to infiltrate the tumor, epithelial cells look much more elongated, perhaps could be more EMT-like state, and maybe these cells are trying to migrate from the periphery of the tumor, but these immune cells are unable to infiltrate, and there is much less trauma here. So our lab trying to understand the various component of the tumor tissue, mainly focusing on liver cancer and endometrial cancer. And in today's talk, I'll be focusing only on the liver cancer, hepatocellular carcinoma. And we're trying to understand the various components in terms of components which are shared between a fetal liver tissue and tumor tissue, and what does it do to the tumor microenvironment. Also, we are trying to understand the signaling pathways which are conserved between a growing embryonic tissue such as fetal liver and which are reappear in the cancer. Maybe we can target these pathways for better therapeutic outcomes. And finally, we want to understand how can we target these conserved interactions between a embryonic tissue and tumor. And for some of the example is by using ongoing clinical trials where they're using antibodies against anti-VEGF and anti-PD-L1. I might not go into too many details about the Oncofetal ecosystem in today's talk, because as I just mentioned, I'm giving another talk and I just don't want to repeat everything. But it suffices to say that we started exploring the Oncofetal ecosystem in late 2017, and early 2020, we published this work where we use a comparative single-cell transcriptomics approach to compare between a fetal tissue and tumor tissue, and we identified some cell types were shared between fetal and tumor tissue, but these cell types were absent in the normal homeostatic tissue, both in the adjacent normal tumor as well as in the healthy normal liver. We also identified which are the key transcription factors might be playing a role in this process, and as well as in early 2020, whatever the tools we had available, we tried to understand the very basic spatial localization of these Oncofetal ecosystem. And our model is something like this, where we show that there's a dividing hepatocyte, which is presented in red here, send the VEGF signaling, which lead to the remodeling of the tumor endothelium. Now, these endothelial cells reprogram macrophages, which are involved in immunosuppressive interaction with the T cell. And as I just mentioned, this is very brief, but if you're interested in understanding more about this, I'll be talking about it in the afternoon. And now, since I moved to Perth early last year, my lab is more interested in understanding what are the clinical implications of this Oncofetal reprogramming and spatial transcriptomics. And in this direction, what we are doing, we are using this single cell atlas and going back into the bulk data, where we are trying to understand what happened in the patient with more or less Oncofetal cells. What are the clinical parameters? Can we understand if having high number of fetal-like cells allow us to predict something about the therapy response? And the short answer is yes. And again, I'll talk about it in the afternoon. So these are some of the approaches we take. We use single-cell RNA-seq and use deconvolution approaches to deconvolute the bulk RNA-seq data. And we also use a similar approach to deconvolute some of the other spatial methods where resolution is not at the single-cell level. And when we were doing these experiments, we found something very interesting, that when we stain the liver tissue and try to sequence different part of the liver tissue in terms of, let's say, we try to punch biopsy different part of the liver tissue, we find that there's something very different between the tumor which is on the periphery compared to the core of the tumor, and we find more fetal-like cells on the periphery. And all these were done using taking a 300 to 500 nanometer big piece of a tumor and then using deconvolution approaches to deconvolute which cell types are present. And when we were doing this experiment, at similar time, Miriam and Maria published this very beautiful study in non-small cell lung carcinoma, where they showed that how the localization of these tissue-resident macrophages, which are in this case also tumor-associated macrophages, influence the tumor cells, right? And so that's where we started getting more excited about this differential localization of the fetal-like cells, why some cells are more into the periphery versus the core, and what does it might be doing to the tumor tissue. And when we were trying to go into more detail into this, we got introduced by Decode, Josh and Peter, and then he introduced us to BC, where we started doing this very interesting approach of taking an entire tumor, because Decode allows us to look into the entire tumor, and then we can ask this question: which oncofetal cells are present in the periphery versus core, and how does it might be influencing the tumor microenvironment? So I'm not going into the detail because Kira and Dan both have already discussed about it in much detail, and I won't be doing justice with that. So I'm just going to skip into the detail of the technology and directly jump into what we are finding in this. So in our very first experiment, we used 2 HCC tissue for this presentation. I'm going to call it HCC1 and HCC3. And we were first of all very pleased to see that the number of genes we had detected in this tissue, and I think this is one of the best I have seen in the liver. And we do a lot of spatial transcriptomics, but looking at 8,000 genes in some of these areas was really good. And I think this is a question is coming again and again in this today's morning session, what kind of details we can get at subcellular resolution. And we had the similar question, and we did this very simple analysis by going into the bin and trying to understand how many genes you can get. Obviously, at a very big bin size, you get a lot of genes, and as you start coming into a smaller bin, you get very less genes. But for example, one of the tissue, even if you go to bin size of 10, which is really small, you still get something in terms of maybe 50 to 100 genes. And I think we have not reached the sequencing saturation in this case. And the next experiment we're going to do is that we're going to sequence it a little bit more to see that if at very low bin size, can we get more number of the genes. So most of the analysis which I'm going to show now is that we have done at the bin size of 25, which is almost 1 or 2 cells. And because this suits what we are doing in terms of looking at cells which are co-localizing with each other and what kind of tumor cells are in this microenvironment. These are some of the common marker of macrophages, endothelial cells, fibroblasts, T cells, and Tregs, etc. And as you can see, they are present in both the tissues. But more importantly, we were interested in some of the genes which are very specific to the oncofetal cells. So we are able to locate these genes at very low resolution. So that was really good, because even if we are detecting, let's say, 300 or 500 genes at very low bin size, we are still able to identify some of the key genes. And this allows us to identify the cell types at almost 1 or 2 cell resolution in this case. So we could identify different oncofetal cells. In this case, I'm showing you the oncofetal cell 1, 2, and 3 based on the gene 1, 2, 3 we know in this case. And this is something we did call network analysis. And again, I'm going to discuss a little bit more about this in the afternoon talk. But what we did this year is that since we know that there are 3 different microenvironmental cells, for example, oncofetal cell 1, 2, and 3, now we are asking the question where these 3 cells come together in the tumor, right? And in that case, we could create some kind of a neighborhood score. And this allows us to understand which part of the tumor has highest concentration of the oncofetal cells compared to the other part. So if you look into HCC3, as is as we can see very nicely that there's a spatialized localization of these oncofetal neighborhood, and it's just mere coincidence that it resembles with the map of the Australia, and maybe there are more oncofetal cells in the western side, just coincidence. But we could validate this by using other approaches, for example, multiplex RNA fish, where we could use 12 different probes to the sum of these markers of the oncofetal cells and microenvironmental cells. And then we can start asking the question, what kind of tumor cells are present? So to summarize this part, StereoSeq allows us to identify oncofetal cells and validate this observation that these cells are present in the periphery of the tumor by looking into the entire tumor. We also, I think I don't have this slide in this talk, but I have in the afternoon talk. We could see that this oncofetal ecozone is associated with EMT-like cells in the HCC, and this is something we are chasing very closely these days. And then we think because quantifying the number of oncofetal ecozones by neighborhood analysis may allow us to identify some tumors which will respond to therapy versus not. And again, I'll have a little bit more about this in the afternoon talk. So I think the idea of this talk was to showcase what we could achieve with Decode and BGI, which were unable to achieve with other existing technologies. And again, I use all the technologies, so every technology has its good and bad side. But BGI, I think, allow us to look into very small resolution and allow us to still detect key genes which are biologically very important. So I would like to thank Decode and BGI for allowing us to collaborate with them and getting this very beautiful data. And I would like to thank my lab members who have analyzed the data, and Liver Cancer Collaborative, which we are very closely working together. So I'll stop here and take any questions. Thank you.
Hi, thanks very much. It was fascinating. You mentioned earlier in your talk that you did deconvolution on spatial data. I guess that must have been Visium. And I would like to have your experience. How does deconvoluting data works for you in comparison to STEomics? Like trying to see whether, again, lots of advantages or what are the pros and cons doing deconvolution versus STEomics?
I think I would refrain myself from talking about comparing one tech versus another because every tech has its own advantage. But I think deconvolution works very well if you have single-cell RNA-seq data from the same tissue, which you may or may not have all the time. If you don't have it, then it's an extra economical burden on the lab. While in BGI, we don't have to do any of these juggleries. So yeah, so I think maybe relatively easier to annotate your cell types without deconvoluting in BGI.
Okay, thank you.
Thanks, and thanks, Marana, for asking a question as well. So I'm not completely dominating the session in the questions. But I've got to be in my bonnet, and this question of how deep do you have to sequence to not have to be able to capture those bins, those small bins. And I was wondering if you had thought about, or if other people have thought about, or if BGI is doing some sort of depletion of the most abundant genes, like ribosomal genes and genes that you are perhaps not so interested in getting that sequencing depth. Just your thoughts, really, would be great.
Yeah, I think it's a great comment, and honestly, I didn't think about it, but I think it's a great comment. And if we can do this, we can actually save some money and get more out of what we spend on these sequencing sites. So yeah.
Yeah, I wonder, even at library prep stage, maybe there's a chance of just having probes or something to get rid of the stuff that you don't want to sequence again and over and over.
Yeah, I agree. And I think so all these experiments, at least the first two experiments, were done at the BGI Shanghai facility. And now we have just got the reagent as part of the beta thing. So we are doing it. And I think it's a great suggestion. We'll try to incorporate it to deplete the ribosomal thing.
Yeah. Good luck with that paper.
Thank you.
Thank you, Uncle, for your wonderful talk. And if there are no more questions from the audience, I would like to, on behalf of BGI and DECO team, I would like to thank for all the speakers for their insightful talk about BGI STOmics technology. And thank you all for attending this workshop. And enjoy your day. And if you have any more questions, we are still around in tea break, and please visit our booth. Thank you all for coming.